This is a production of Cornell University.
Great. Well, good afternoon, everyone. I'm excited to talk to you about my dissertation research today, titled Genomics-assisted breeding of malting barley for New York State. So by the year 2050, it's expected that there will be 9 billion people on this world, and food production will be challenged by increased population, climate change, and land degradation. And that means, well, we're going to need more beer. And that means we need more barley. Barley is a diploid grass that's been the primary ingredient in brewing and distilling for millennia. There are two primary classifications of barley. The first is by how the grains are arranged on the spike, and that's 2-row versus 6-row. The picture on the right shows 2-row barley. And the second is by vernalization requirement. Winter barley is planted in the fall, and spring barley is planted in the spring. I will primarily be talking about 2-row spring malting barley today. In North America, most barley production is in the West. In the United States, in Montana, Idaho, and North Dakota, and in Canada, in the prairie provinces of Alberta, Saskatchewan, and Manitoba. However, New York State has a strong barley heritage. In the mid-1800s, New York was a barley production powerhouse, with most acreage focused in the western part of the state along the Erie Canal. This fed large malt houses and breweries in Buffalo, but continued westward agricultural expansion, as well as a series of tariffs with Canada, killed malting barley production in the late 1800s. And barley has since languished as a major crop in New York since then. But that's beginning to change. In 2012, the New York State Legislature passed the Farm Brewery Bill. This legislation created a new class of craft brewery in New York. Farm breweries receive economic incentives and the fun tagline of being a farm brewery if they use locally grown grain and hops in their beers. Now, in 2012, there was no malting barley production in New York. So Cornell was tasked with increasing malting barley acreage. But so far, this Farm Brewery Bill has already led to massive growth in the number of breweries in New York State. There are now over 400 breweries in New York, which places us in the top 5 in terms of number of breweries per state. So in 2013, Mark started trialing barley varieties from Western North America and Europe to try and find varieties that would suit local production, as well as craft malting and brewing in the state. But the typically wet and humid growing season that can be rather unpredictable in New York introduced a lot of challenges in barley production. And these were agronomic, disease, and quality challenges. So because none of the varieties that we trialed were really a good fit for us, we decided to initiate our own breeding program for 2-row spring malting barley so that we could service the needs of farm breweries within New York State. And we named this breeding project the Born, Bred, and Brewed in New York project. Now, the Farm Brewery Bill has pretty strict quotas for how much grain is needed to be a farm brewery. And by 2024, that quota for farm breweries is 90% of their grain bill. So that means that we needed to act quickly in order to release a superior malting barley variety that met the needs of local growers and end users. Now, until recently, the only selectable marker that plant breeders used was a can of spray paint. And spray paint still has its uses in the breeding nursery, but the advent of molecular markers has radically changed how we think about breeding and structure our breeding programs. The term marker-assisted breeding has been used for the past 2 decades or so to refer to methods and technologies to implement molecular markers into the breeding program. I prefer the term genomics-assisted breeding because in the 2020s and the 2010s, we now have access to genome-wide molecular resources that really expand the range of options available to breeding programs. But the question is, how do we integrate these resources into a breeding program to increase our efficiency and accuracy? So in barley, we're likely to have some very high quality genomics resources. These include a high quality reference genome, a recent 20 accession pan genome, and a 50K SNP chip that is built upon previous smaller SNP chips for nice continuity. So my interest is to use these genomics technologies to improve quantitative analysis that supports breeding decision in our brand new barley breeding program. So I'll be describing a few different methods of how I've employed genome-wide association, improving genotype and environment interaction modeling, and genomic prediction and selection to aid our efforts to support craft brewing in New York. So this talk will have three main parts. The first will be genetic mapping of pre-harvest sprouting and seed dormancy. The second will be modeling environmental sensitivity of pre-harvest sprouting. And the third, we'll take a look at our efforts to apply genomic prediction and selection in a new barley breeding program. We will start with pre-harvest sprouting. So I've used the term malting several times to describe the sort of barley we're working with. Barley can also be used for human food and animal feed, but the highest value is for malting. But what actually is malting? Cereal grains store energy in the form of starch, but germinating seedlings need simpler sugars in order to grow. Yeast also need to eat simpler sugars to make alcohol during the fermentation process. So malting takes advantage of the natural germination process to prime the conversion of starch to sugar. So malting has three basic steps. First, the grain is soaked or steeped to initiate germination. Germination is then allowed to proceed for 3 to 4 days. And then germination is arrested in the kilning stage where a dry heat is applied. And that also helps preserve enzyme activity and develop color and flavor. So why do we care about enzyme activity? Amylases are the enzymes that break down starch into simpler sugars. And that actual conversion of starch to sugar does not happen until the first step of the brewing process. But malting kind of primes the grain in order to be able to make alcohol efficiently. There's a protective matrix of protein and beta-glucan that surrounds the small starch molecules within the barley grain. And during the germination process, this matrix is broken down and amylase enzymes are activated. This is called modification, and uniform rapid germination of barley seeds in the malt house is needed for efficient, complete modification. Malting quality is actually a suite of 10 to 12 quantitatively inherited traits that are all related to grain modification and grain chemistry, protein and starch. And as you might expect, phenotyping is slow, expensive, and requires a lot of grain, much like other quality traits. Seed dormancy is the inability of a seed to germinate under otherwise favorable conditions. And seed dormancy is a very important consideration in malting. Because malt houses require germination to make malt, they require non-dormant grain. And in the context of malting, there are two primary phenotypes that we're interested in when it comes to seed dormancy. The first is germination energy. This is simply a measure of seed dormancy that's measured as the germination percent over 3 days, with a higher germination energy indicating no dormancy. And germination index is a measure of germination rate over 3 days, with a higher germination index implying a higher germination rate. So these two traits are important for malting, but in New York, for our germplasm and for malt houses in general, we have a limited understanding of how germination rate changes over time. We're interested in this because germination rate has been positively correlated with some malting quality traits. It may be a useful proxy for more cost-efficient and time-efficient selection for malting quality traits. Seed dormancy duration is also important. One, because malt houses don't want to sit on dormant grain because they can't malt it. And two, because of its connection with pre-harvest sprouting. In varieties with very low seed dormancy, wet harvest conditions can sometimes induce germination on the spike in the field. And this can completely ruin barley quality. Considering that wet harvest conditions are frequent, if not the norm in New York, this is a major concern for barley production in the state. Pre-harvest sprouting is an expensive phenotype. It's very time sensitive and labor intensive, and our phenotyping method requires hand sampling at a specific maturity stage, after ripening or drying for 3 days, and then setting up on styrofoam trays that we put in a mist chamber in our greenhouse. These are then misted for 3 days to induce sprouting, and we score them on a 0 to 9 scale. Here I shows only 0 to 7, but 0, there's no root or shoot emergence, and 7, you can see many green shoots and many roots, which indicates severe sprouting. Pre-harvest sprouting is highly heritable within a year, which helps breeders, but some varieties have substantial across-year variation. The biggest challenge with pre-harvest sprouting is that it's often negatively correlated with malting quality. This is not surprising, considering that high seed dormancy is generally connected with pre-harvest sprouting resistance, and high seed dormancy is also associated with slow germination and poor malting quality. So our goal for craft breweries in New York is to try and see if we can select for pre-harvest sprouting resistance and good malting quality. Because of the economic importance of these traits, a number of genetic mapping studies have been conducted in diverse barley populations for these traits, and 2 consistently detected QTL on chromosome 5H have been detected for both pre-harvest sprouting and seed dormancy. The first locus, seed dormancy 1 or SD1, there has been an alanine aminotransferase cloned at this locus. There's also been an ALA-AT1 associated with pre-harvest sprouting in wheat. And the second locus, seed dormancy 2, is a MAP kinase 3 has been cloned at this locus. And MKK3 has also been associated with pre-harvest sprouting in wheat and rice. So these 2 large effects, unlinked genes on chromosome 5H, have already been cloned. Why are we trying to map these? At SD2, malting quality and pre-harvest sprouting QTL are known to co-locate and are negatively correlated. But it's not known if this correlation is due to a cluster of tightly linked genes with separate effects on malting quality and pre-harvest sprouting, or if it's pleiotropic effects of a single gene. Intriguingly, in the SD2 region, there is a gibberellin 20 oxidase 1 gene. Gibberellin 20 oxidase is involved in active gibberellin biosynthesis, and gibberellin is a major promoter of seed germination. GA20OX1 is known to be expressed in germinating barley seeds and has also been associated with pre-harvest sprouting in wheat and rice. If there are variants at GA20OX1 and MKK3, and they both have effects on malting quality and or pre-harvest sprouting, it may be possible to decouple that tight linkage through breeding and select for good pre-harvest sprouting resistance and good malting quality. So our objective was to identify and assess temporal interactions of pre-harvest sprouting and seed dormancy QTL. So before I talk about methods, I first want to acknowledge the substantial contributions in this section from my colleague, Travis Rooney. So our approach was to use genome-wide association for pre-harvest sprouting and germination traits using 3 data sets. The first was a panel of 400 Cornell 2-row spring malting barley breeding lines. And the second was a population composed of 2 sources. The first was about 80 European landrace successions curated by the John Ennis Center from the UK, so abbreviated JIC. And we added about 20 hulless or naked barley lines from my colleague Carl Kunze's research for a total of 108 lines in that subpopulation. The CU in the JIC material was grown in 2019 and 2020 and phenotype for pre-harvest sprouting, germination energy, and germination index, although JIC was only phenotyped for GE and GI in 2020. Both of these populations were genotyped with the 50K SNP chip, and the principal component plot on the right gives an indication of how these populations are related. This big blue blob on the right shows that the CU breeding lines are closely related, while the red JIC material and the black naked material are substantially more diverse. Our third data set was the result of a collaborative project with the University of Minnesota. This is part of a spring 2-row multi-environment trial. It's abbreviated S2MET. And these are 200 lines that were composed of breeding lines from Western North American breeding programs. This is phenotyped for pre-harvest sprouting only in 2015 and 2016 and has GBS genotypes. So seed germination phenotyping is not a trivial task. It's very labor intensive and requires sampling of a large amount of spikes just after physiological maturity and hand threshing of all those spikes. because mechanical threshing can damage the seed coat, which will impact seed dormancy and germination rates. Once we hand threshed all of our samples, we froze those samples at -20°C so we could assay at the same maturity stage. And then we hand counted and plated 30 kernels per sample and hand counted the number of germinated kernels each day for 3 days to measure GE and GI. Because we were interested in how GE and GI changed over time, we assayed at multiple after ripening time points in each year, ranging from about 1 week after physiological maturity to about 3 months. And we increased our sampling frequency in 2020 to 6 time points. So because we knew that we were interested in 3 specific seed dormancy genes, we collaborated with the USDA Cereal Crops Research Unit and Jason Walling's group to Sanger sequence panels of released cultivars and breeding lines, winter and spring material, for alanine aminotransferase, GA20OX1, and MKK3. And based on these Sanger sequencing results, to support and refine our genome-wide association mapping, I developed KASP assays for each of these 3 genes. So I was able to develop a KASP marker that amplified the causal variants in ALA-AT1, which is very useful. When we Sanger sequenced GA20OX1, we detected 4 different alleles of GA20OX1, but no non-synonymous mutations in the exons. And none of the alleles were obviously associated with pre-harvest sprouting variation. Nevertheless, I developed a KASP assay that amplified a 5' UTR SNP in GA20OX1 that differentiated between the 2 most common alleles. I then developed 2 markers to genotype MKK3. And this is because a paper published by Vetch et al. From Montana State, and also in collaboration with Jason Walling's group, detected 3 different MKK3 alleles in modern North American breeding germplasm. Vetch et al. Only associated the N-star MKK3 allele, shown in the top row here, with pre-harvest sprouting susceptibility, but I was interested in the effects of these second 2 MKK3 alleles. So I developed 1 marker, MKK3E165Q, to amplify the mutation in exon 3 that differentiates the N-star allele. And I modified a 50K SNP chip marker that tags the linked variants from exon 7 and exon 8 to differentiate the next 2 alleles. So going forward, it's important to remember that these 2 markers differentiate 3 alleles of MKK3. So I conducted genome-wide association analysis using a multi-locus mixed model in GAPIT. I added 2 principal components to confer population structure. The CU and JIC material were analyzed together because they were grown in the same environment, and the S2MET material was analyzed separately. And this analysis helped validate a lot of our KASP markers, which is exciting. So this plot at the right is a Manhattan plot summary to kind of put all these time points and all these traits into one hopefully easy to read plot. And we found that alanine aminotransferase, the causal variance, was associated with germination energy and germination index across time points. So that's shown in this peak here in the SDE1 region. The MKK3E165Q marker was the only marker associated with pre-harvest sprouting, but was also associated with GE and GI across time points. And these are shown in the squares and are highly significant. However, we also detected marker trait associations for the second MKK3 marker for GE and GI across time points. So this data suggests that each of the 3 MKK3 alleles has distinct effects on germination energy and germination index. We did not detect any marker trait associations for the GA20OX1 marker for any trait at any time point. And I should mention that these results are just for the CU-JIC material, but we had similar results in the S2MET. So showing that data a little bit differently, I grouped the phenotypes for pre-harvest sprouting for the CU-JIC population by their large effect seed dormancy haplotype. So the haplotype is defined as the combination of alleles at alanine aminotransferase, GA20OX1, and MKK3, with N signifying non-dormant and D signifying dormant. So here we can see that haplotypes that have the highly non-dormant MKK3 and star allele in red and orange are pre-harvest sprouting susceptible. And in this CUJIC population, the other two MKK3 alleles are all pretty pre-harvest sprouting resistant. We also don't see any effects of ALA-AT or GA20-OX1 on pre-harvest sprouting. When we look at the S2-MET data, we do see effects on pre-harvest sprouting of each of the MKK3 alleles. So again, in red and orange, the highly non-dormant and star allele is very pre-harvest sprouting susceptible. In blue, the weakly non-dormant N allele is somewhat pre-harvest sprouting susceptible, and there's more dormancy conferred by the dormant MKK3D allele. And once again, GA20-OX1 has no effect on pre-harvest sprouting here. So the effects of the MKK3 alleles become a little bit more clear when we look at germination index and germination energy and how those change over time in the CUJIC material. So on the top, we have germination index and germination energies on the bottom. And each of these facets show these germination traits at 6 days after maturity, 48 days after maturity, and 111 days after maturity. And similar to pre-harvest sprouting, we see that the highly non-dormant and star allele has high germination index, so it sprouts quickly across all time points, and has no primary seed dormancy. And that, not surprisingly, lasts throughout the after ripening period. Whereas the dormant allele has a much slower germination rate at all time points compared to the most non-dormant allele, and has substantially higher primary seed dormancy that persists at least a month and a half after maturity. Note though that the effect of the dormant MKK3 allele is conditioned by the state of alanine aminotransferase 1. Dormant ALA-AT1 combined with the dormant MKK3D provides very high seed dormancy that persists the longest of any haplotype. This also is combined with a very slow germination rate. So these are not good malting lines because they germinate too slow and are too dormant. MKK3N, though, has some favorable haplotypes for malting and brewing, especially the combination of non-dormant alanine aminotransferase and the weakly non-dormant MKK3. These have very low primary seed dormancy and a good germination rate that quickly approaches the germination rate of the most non-dormant and star alleles. They also have pre-harvest sprouting resistance. So these data suggest that the 3 MKK3 alleles are responsible for effects of pre-harvest sprouting and seed germination traits at SD2 and not gibberellin 20 oxidase 1. And I want to highlight this a little bit more because I think this is one of the more interesting broader implications for this work and how we interpret GWA results. So before I added the KASP assays, I just had my 50K marker set, and I had about 14,000 markers. It was a good dense set in this region. But because of the highly structured nature of the Cornell breeding material, the marker and closest linkage with or the highest linkage with the N star allele happened to be in this LD block that conveniently surrounded GA20-OX1. And there is a separate LD block that surrounded MKK3. So I was convinced for several months, actually, that we did have variants at GA20-OX1 and MKK3. But until we complete our Sanger sequencing of GA20-OX1 and developed KASP assays for both of these markers, and then adding the more diverse material from the JIC to the GWA analysis, then I was able to pick these genetic signals apart a little bit and realize that it was actually three alleles from 2 markers at MKK3. And both of these markers were in different minor allele frequency, which is why we could detect them in the first place. So for anybody else trying to do genetic mapping, especially if you have a population that has extended linkage disequilibrium, limited or no recombination between 2 loci of interest, and low marker density, which is the case for many previously published biparental populations, you might have this illusion of 2 separate loci that could possibly be multiple alleles of a single gene. So now that we know the primary genetic effects on pre-harvest sprouting, I was interested in trying to answer the question of why some varieties are so variable in pre-harvest sprouting across years, while others are very pre-harvest sprouting stable. So to do that, a quick primer on seed germination physiology will help us begin. Seed germination is a complicated physiological process conditioned by interactions between many plant hormones and environmental stimuli, but the two most important hormones are abscisic acid, abbreviated ABA, and gibberellic acid, abbreviated GA. ABA promotes seed dormancy, while GA promotes seed germination. And the balance of those two promotes whether the seed will germinate or be dormant given favorable conditions. There are many genes that impact the biosynthesis and signaling of both of these hormones, but of interest to us today is that temperature is known to have an effect on ABA biosynthesis and signaling in barley. Temperature is also known to have an effect on pre-harvest sprouting in barley and other cereal grains, with higher temperatures increasing pre-harvest sprouting susceptibility and decreasing ABA levels. So classically in plant breeding, the way that we try to determine the factors that determine how a plant looks, so in this case, barley pre-harvest sprouting, we partition that into the impact of genetics, G, the environment that that plant grew up in, E, and the interaction of those two factors, genotype by environment interaction. So I decided to take a QTL by E modeling approach to try and explain annual variation in pre-harvest sprouting. I did this because we had information on large effect seed dormancy QTL, so I could fit those as their own effect, and then model any residual quantitative background genetic variants as G. For environment, I divided it into physical year location combinations and decided to model environmental covariates as well. These are things like temperature and precipitation that might affect pre-harvest sprouting susceptibility. And then G by E term is just the interaction of each of these factors. So I modeled my approach after Malisetti et al. For QTL by E modeling. I fit main effects for physical environment, QTL, and genotype, residual genotype, and then fit interactions between all these factors. The bolded terms were fit as random effects, and all these models were fit in ASRMLR. And I was able to do this analysis because we had 5 to 6 years of historic pre-harvest sprouting data from our breeding programs. So I modeled spring and winter pre-harvest sprouting separately. Notice that our spring data set is larger. That's because we've invested more time and effort in spring barley breeding to this point. And I used 5 environmental covariates. The first was maximum temperature, and then minimum temperature, average temperature, temperature range, and total precipitation across the grain fill period. Grain fill being defined as flowering time to pre-harvest sprouting sampling dates. Notice that temperatures are higher for the spring-grown grains and encompass a wider range in temperatures. We have similar precipitation patterns. The temperature data, or the weather data, was representative of the last 30 years in Ithaca, New York. Because the spring data set was larger, we also were able to sample more seed dormancy haplotypes. And again, these haplotypes are defined as the combination of alleles at ALA-AT, GA20OX1, and MKK3 in order. Most importantly to note here, the spring data set had observations for all 3 MKK3 alleles, for the N star, the N, and then the dormant, while the winter data set just had the highly non-dormant N star and the dormant alleles. So based on part 1, we learned that pre-harvest sprouting is a function of genotype, but these results strongly suggested that environment and G by E have large impacts as well. Best model fit was determined by AIC, and total precipitation was the best model fit for the spring model. Temperature range was the best fit for winter, but somewhat more interpretable, maximum temperature was a good fit for both. The plot on the right shows the percent variance explained by each of the random model terms, with red being the residual background quantitative genetic variance, yellow, which is not really detectable on the winters, but is this thin line here in the springs. This is genotype by environmental covariate interaction, so genotype by temperature. Blue is genotype by physical environment interaction. Green is QTL by physical environment interaction. And then black is residual error. So for all temperature covariates, or all weather covariates in spring and winter data sets, the fixed environment and QTL effects were always significant. But in the spring, we had higher proportions of genetic and genotype by environmental covariate interaction variants. And this is possibly just simply a function of having more entries in the spring data set, 61 as opposed to about 25 in the winters. Whereas the winters had substantially higher QTL by physical environment interaction. And this may have been a result of some material being pretty unadapted to cold New York winters and having severe winter kill or very bad disease problems in some years, which could impact flowering time and the grain fill duration. So if we look at this a little bit closer, we can see that there is pre-harvest sprouting temperature sensitivity that varies within and between seed dormancy haplotypes. So these plots show environmental sensitivity for the winter and the spring data set, specifically for maximum high temperature during grain fill. And these top two plots on the winters are the highly non-dormant MKK3 and star allele. And we can see, as we learned in part one, they have a high baseline pre-harvest sprouting, but that pre-harvest sprouting increases as temperature increases. We see a similar pattern for both of the spring and star allele classes. While for the dormant allele, we have high pre-harvest sprouting resistance that really stays very stable as temperature increases. And we see similar results for each of the dormant MKK3 groups in the springs. And in some cases, pre-harvest sprouting risk actually decreases a bit as temperature goes up. Now, interestingly, for the springs, the non-dormant MKK3 allele generally starts as pre-harvest sprouting resistant, but is very temperature sensitive, with many lines becoming susceptible as temperature increases 4 to 5 degrees. Also note that for the springs especially, there's a large amount of variation in intercept. And I forgot to mention that the black lines here are the main QTL by environmental covariate interactions. So these are the main QTL by temperature interactions. Variation in intercept is due to residual genetic effects. So these are individual effects of varieties. And then any variation in slope is unique variation in a variety by temperature sensitivity. So we actually have some crossover interactions for the spring material. This is of interest because it suggests that within haplotype group, there is some quantitative variation remaining for pre-harvest sprouting resistance, which may be useful for breeding efforts. These results were further supported by our old friend, the S2MET data set. And conveniently for me, the S2MET was grown in a very cool and wet year in Ithaca in 2015 and a hot drought year in 2016. And we can see that the non-dormant, highly non-dormant and star allele in red was pre-harvest sprouting susceptible in both environmental conditions, whereas the other two MKK3 alleles were dormant in 2015 and became non-dormant at different rates in 2016. So we had more temperature sensitivity in the blue N allele and less temperature sensitivity in the gray dormant allele. I don't have time to get too much into the weeds with the physiology of ABA temperature sensitivity and how MKK3 is interacting with ABA. But based on what we do know about the action of MKK3 and ABA, the temperature sensitivity of MKK3 is likely related to ABA sensitivity. Okay, so simply, MKK3 N star has low ABA sensitivity, MKK3N has moderate ABA sensitivity, and MKK3D likely is highly ABA sensitive. Okay, so what are the breeding implications of these first two sections? And just kind of the take-home notes here. So importantly, at the ST2 locus, pleiotropic effects from pleiotropic effects of that locus are strongly suggested to be caused by multiple MKK3 alleles, not tight linkage with GA20OX1 and MKK3. And there are no other strong candidates in the ST2 region for seed dormancy or pre-harvest sprouting. They could still be there, but there are none that seem likely. GA20RX1 was by far the best candidate. And this should help breeding efforts because the KASP markers for these two genes should help breeders select an appropriate seed dormancy level for their target population of environments, as well as their target malting quality profile. And maybe if we have time at the very end, I can show a slide with some preliminary malting quality mapping results and the S2-MET with the MKK3 data. But it does seem that there are pleiotropic effects of the highly non-dormant and star allele on some, but not all malting quality traits. And those malting quality traits are more specifically associated with a malting quality profile preferred by industrial malting and brewing, not craft malting and brewing. So this is promising because it suggests that selection for pre-harvest sprouting resistance, short primary seed dormancy, high germination index, and good craft malting quality does seem possible for us in New York and other areas trying to grow good craft malt. And then importantly for production and for breeding efforts, MKK3 alleles have varying temperature stability. For breeding, this means that breeders need to continue to test within MKK3 allele to see how stable those alleles are across years. And these models could also be used as a predictive tool for growers to forecast if a particular variety will sprout given the weather up to harvest time. So mostly moving away from seed dormancy and pre-harvest sprouting, I want to close by describing some of our efforts to apply genomic prediction and selection within our new malting barley breeding program. So for thousands of years, plant breeders have successfully selected on a plant's phenotype, so just how the plant looks, and that has allowed us to make genetic gain and form agriculture as we know it. But the introduction of molecular markers means that we can now select much more efficiently and much more accurately than purely phenotypic selection. And much ink has been spilled within our lab, other labs at Cornell, and now research groups globally on the promise and applications of genomic prediction and genomic selection. I'll just qualify here that genomic prediction is referring to actual prediction of plant performance based on genotypes, whereas genomic selection is using genomic predictions within an actual breeding framework. So in a nutshell, genomic prediction, we're able to capture the relationship between individuals based on their genotypic data that we assess with molecular markers in a genotype matrix. And there are many possible benefits of genomic prediction, but three of the big ones are potential to predict unphenotyped individuals and unphenotyped locations, which can save a lot of time and money, increase breeding cycle time, which may increase rates of genetic gain, and improve our selection accuracy within families. So the key for genomic prediction success is having a good training population. So this cartoon on the right is a very simplified version of how genomic prediction is structured. You have a representative training population, which is just different breeding lines or different types of plants that you genotype with molecular markers and phenotype for your traits of interest. Plug that data into a statistical model, and that predicts your target test population of plants. And these test plants are only genotyped. So the key to good genomic prediction relies in having high relationship between your training and your test sets, a good sized training population with dense enough markers to capture all the haplotype blocks, and sufficient phenotypic variation for your trait of interest. So given that we were trying to breed as quickly as possible, and that, let's be honest, I'm in Mark's lab and we've done a lot of genomic prediction work, and I probably didn't have a choice, but I wanted to learn how to do this. So our method to breed malting barley quickly was to use genomic prediction and selection. And we decided to structure our training population as a connected half SIB population. So very simply, this population involves the crossing of multiple parents to a common parent. So for very, very structured, connected population. This gives us a couple of potential advantages for increasing prediction accuracy. The first is that we can leverage across and within family variation. It gives us high statistical power for genome-wide association analysis. And we're not interested in understanding the genetic architecture per se of the traits, but is more to detect large effect loci that could be added to the prediction model to further increase prediction accuracy. It also allows us to efficiently genotype, where we can genotype the parents at a high plex and then impute the progeny that were genotyped at a lower plex. And this population naturally has high relatedness, which is bound to be good for prediction accuracy, and allows us to ask some interesting prediction questions about combining and cross and within family training sets. So again, this is the structure that we actually used. AAC Synergy is the most popular variety that we have now in New York, but it has some disease issues and is very pre-harvest sprouting susceptible. So this is our kind of elite malting background, and these are other malting barley variety parents that we crossed to Synergy. So the training population that I developed ended up being about 1,300 lines in those 7 families. And these were planted in 2 locations in Ithaca in 2017, and meter-long head rows as shown here. And these were phenotyped for agronomic traits, quality traits, and disease traits, and genotyped with about 6,000 GBS markers. And single trait GWA models were fit for each of these traits. I explored a range of prediction accuracy and cross-validation questions, but I want to talk about the actual genomic selection process. So if you're interested in those were published in Crop Science early in 2020. Okay, so over the course of 3 years, I was able to complete 2 cycles of genomic selection. And I actually selected on base selection index values that were calculated from GEBVs, calculated from those single trait GWA models. So agronomic traits like heading date and height were lightly weighted because we weren't sure what our target range for those traits were yet. And traits like pre-harvest sprouting and spot blotch were heavily weighted. So this diagram on the right is a little cartoon of the selection scheme. This shows the training population in each of the 7 families. I dropped 2 of the families from selection because they were highly related to synergy and had low variance for our traits of interest. So based on selection index values, I selected the top 2 lines from each of the remaining families and intermated them in all the possible half SIB matings. So that means that the 2 SC lines were not intermated to try and stave off severe inbreeding, but were intermated with all of their half SIBs. So these selections are made using genomic prediction and also just phenotypic selection. So that resulted in 211 phenotypically selected lines and about 370 cycle 1 genomic lines. Cycle 1 genomic lines were genotyped at the F2 stage with a subset of GBS markers that were converted to an LGC SNP assay. And the predicted top 15 lines were then intermated to create cycle 2G. So to evaluate gain from selection, I needed to evaluate these populations in the same field. Training population 2017 was far too large to plant again. So I randomly sampled 15 lines from each family to perform a proxy training population that I called C0. 100 C1 phenotypic lines were randomly selected. The top 92 C1G lines were selected, and then all 100 C2G lines were selected. So these cycles were simultaneously grown in 2 locations each in 2019 and 2020. They were phenotyped for heading date, height, pre-harvest sprouting, and then spot blotch phenotypes were taken in the greenhouse for seedling assays. And all the correlated germination traits that I talked about in section 1, that was taken from this experiment in both years. And these were phenotyped or genotyped with about 21,000 markers to look at inbreeding. So I compared realized versus expected genetic gain for each of the traits and for the overall selection index. And expected gain for component traits within a selection index was calculated with this equation, where G and P are the respective genotypic and phenotypic covariance matrices. W is a vector of trait weights, and I is a selection intensity. So this figure here shows realized versus expected gain across cycles. Pink is the realized genetic gain, and blue is expected genetic gain. And the genomic selection cycles are shown in solid lines, while the dashed lines represent phenotypic selection. So we did have gain for pre-harvest sprouting and overall selection index with small gains for heading date and height. But even though spot blotch was heavily weighted, there was not gain for spot blotch. And my best guess as to why that happened is that phenotyping for spot blotch in the training population was rather difficult due to confounding of early leaf senescence with disease symptoms. And expected and observed gain were pretty similar from cycle 0 to cycle 1 for each trait, but were substantially different for between cycle 1 and cycle 2. And this may have been due to difficulty estimating the genotypic and phenotypic covariance matrices from cycle 1 to estimate cycle 2. One of the primary concerns about aggressive use of genomic selection is that it can rapidly deplete genetic variants and increase inbreeding compared to phenotypic selection. So we observed a reduction in genetic variants for pre-harvest sprouting with genomic selection cycles in blue compared to the phenotypic selection in yellow. But that's also because we actually had gain from selection to pre-harvest sprouting for genomic selection compared to pre-harvest sprout or the phenotypic selection. But for the lightly weighted traits, heading date and height, we really did not deplete our genetic variants very much. And this gives me some hope that we also didn't deplete genetic variants for yield, which we have not been able to measure on these traits yet. We did see that average genomic relationship and monomorphic marker percentage increased with 2 cycles of genomic selection compared to 1 cycle of phenotypic selection, but the inbreeding could have been much worse. I mentioned earlier that I purposely avoided full SIB mating. And if I were, based on simulation, if I were to have only selected the top 10 selection and predicted selection index value lines within each cycle and intermated those, average inbreeding after 2 cycles of GS would have been at least 0.55 instead of 0.41. So again, taking a step back a little bit and looking, what does this project tell us about applied genomic selection? I think the biggest question for us that is still under evaluation, because we don't have all the data yet, is was GS worth the cost in our breeding program? As I mentioned, because I had to graduate, we grew the gain from selection trial in head rows, and we didn't have enough seed to grow them in yield trial plots. So we don't know what the yield is on these yet, and we don't yet know what the malting quality is on these lines, although Jason Walling's group at CCRU is currently working on us with getting those phenotyped. So I think, if we did not deplete variation for malting quality and yield, I think this was a successful approach, although the lack of gain for spot blotch was discouraging. And given what we now know about the genetic architecture of pre-harvest sprouting in our program, marker-assisted selection, if we were only selecting on pre-harvest sprouting, may be more useful. But the selection index does seem to potentially be useful for gain for component and overall selection index, component traits and selection index. So I mentioned some advantages of using a connected half-SIB training population earlier, but I want to bring up a couple of potential consequences to think about as well. The first is founder diversity. A couple of the parents that we selected were very closely related to each other, and that limited our prediction accuracy within those families and limited genetic variation. So if I were to do this again, I would have probably selected more founders and had smaller family sizes, and that would have helped us manage inbreeding a little bit. Although within this connected half-SIB population, you are inherently going to have inbreeding because everything shares a single parent initially. But the importance of these first 2 factors does depend on your short versus long-term breeding goals. We knew that we had a very short-term focus from the get-go. We were really focused on 5 and then maybe 10 years out. But using this, so for a very intense, aggressive selection program that needs to get a variety out quickly, this might be a useful approach. But for longer-term sustained gain, we probably need some more careful evaluation. And lastly, is the statistical power afforded by the connected hapsid population, we can do GWA, but is that worth it? Because most of the time breeders are not really interested in the underlying genetic architecture per se. But I think for something like disease resistance, this could actually be quite useful because knowing if you have a limited number of large effect disease resistance loci in your breeding program would be useful to manage long-term evolution of disease resistance. So while this genomic selection program was progressing, I was also managing our 2-row spring malting barley breeding program. And as Mark mentioned in the introduction, we've rapidly progressed this using a combination of genomic prediction and winter nursery to go from first crosses in 2016 to foundation seed and on-farm trials in 2021. And Mark also let the cat out of the bag a little bit, but this led to the release of the first ever 2-row spring malting barley variety from Cornell, which was christened Excelsior Gold by the New York malting barley community. Excelsior is the state motto of New York, and gold is the color of barley and the color of beer. So Excelsior Gold has yield comparable to our agronomic checks with good foliar disease resistance and fusarium head blight resistance, which is a big problem in New York. And importantly, it has good pre-harvest sprouting resistance. It actually has the MKK3 D allele, if you're curious, and a short post-harvest seed dormancy period with a good germination rate and acceptable malting quality. So with that, I have some acknowledgements. I first really want to thank New York Agriculture and Markets. They have been the primary sponsor of my PhD research, so very grateful for their support of our research in malting barley in New York. A lot of the seed dormancy work was sponsored by the American Malting Barley Association and the Brewers Association, and a grant from the Cornell Center for Technology Licensing helped us push Excelsior Gold out the door. I also really want to highlight the contributions of the New York malting barley community at large. It's been a really fun group of people to work with and to learn from in my time here. They're very passionate about their craft and their trade, and they've been really interested in what we're doing at Cornell and very supportive of our research. And I especially want to thank Andrea Stanley, Ted Hawley, and Dennis Nessel for their contributions and collaborations to our research. So in terms of specific people, I first want to thank my committee, Mark Sorrells, Gary Bergstrom, and Jessica Rudkowski. They've been great to work with and have really supported me well this entire time. Special shout out to Jessica for sticking with me through several career moves. So thank you very much, Jessica. Your input is always, always appreciated. The Yannick Sorrells group, specifically, I want to start with David Bencher, Amy Fox, and James Tanaka, our technicians. We would not be able to do anything in Sorrells lab if not for them. And they make coming to work a lot of fun and a great joy, especially during summer field season. So I'll miss the three of you quite a bit. I've really been blessed to have fantastic lab mates the entire time I've been here. I want to first thank former colleagues, Nick San Antonio and Lynn Veenstra, for being patient with me and answering many, many questions my first few years. So I've tried to return the favor once I was a senior grad student, but big thank you to both of you. Chantel Martinez is a former postdoc who contributed a lot to seed dormancy work, and Carl Kunze and Travis Rooney especially have collaborated a lot on all the barley breeding and the seed dormancy work. At USDA's Cereal Crops Research Unit, Jason Walling and his technician Leslie Zalapa have been excellent collaborators. Jen Starr and Gary Bergstrom's lab helped me a lot with all the spot blotch inoculum prep and phenotyping. At Hartwick College in Oneonta, New York, the Craft Food and Beverage Center, Aaron McLeod has been just a tremendous supporter of the farm brewery activity and malting and barley in New York in general. And he's taught me an incredible amount about malting and malting quality. And I hope that we've been able to teach you a little bit about barley breeding, Aaron. So thanks for all of your help. And Christopher Redoubt at the John Ennis Center was the collaborator on the GIC material. And a few miscellaneous thanks. Chris Hernandez has been my housemate for the last 6 years, the entire time I've been here. And I've learned more from him about plant breeding than anybody else or any seminar. So thank you to Chris, and thanks for putting up with me for so long. To Synapsis, the plant breeding graduate students, you've been a great source of friendship and ideas, and I really look forward to networking and collaborating with you for my whole career. The Cornell Catholic community has been a great source of fellowship and support in my time here. And then lastly, but very importantly, my family. has always very much supported my education through this entire process. So thank you very, very much for your love and support. And lastly, I just want to say that I've been really, really thankful to have this opportunity to study plant breeding at Cornell. It was my dream grad school, and it's been a great place to conduct my dissertation research. But it's given me the opportunity to explore around the world, around the country, and within New York. So my only advice to current grad students is get out of Ithaca, because there's a lot of cool stuff in New York, and it's worth exploring. And I would be happy to hear any questions and comments. So thanks very much for your time.
Thank you very much, Dan. We're running a little bit long, but if people could take a few more minutes to ask some questions, I'm sure Dan will be glad to see if he can answer those for you.
Okay, so I see one question from the Muir Lab. You generate a lot of data. How do you deal with the data management challenge? That's a great question, and I'll admit that we did not do a great job of managing data beforehand. I think that's something as a lab we probably still need to work on, but my colleague Travis Rooney pushed me into using GitHub. So we've been using GitHub to manage and share our data and code a little bit better. So we're working on that, but that's definitely still an active area. I will be uploading a lot of this data to the T3 breed base, but that hasn't happened yet.
Well, that's probably something I could do better as an advisor as well.
It's a big job.
Any other questions from anybody?
Yeah, I have a question.
Yeah, go ahead.
Yeah. Yeah. So have you looked into the candidate genes they involved in ABA or GA biosynthesis or signaling, and look into their possible impact on C dominancy in barley?
Yeah, that's a good question. So there has been a lot of work done in barley looking at things like ABA hydroxylase and the NCED genes, which are also involved in ABA metabolism. I did not look at expression of any of those genes specifically, but I think to get a really good, but I have a feeling that some of those genes might be underlying some of that background genetic variation for temperature sensitivity, because those genes are helping to determine how much abscisic acid is in the grain at any given time. So I didn't look into that specifically, but I think that's definitely an opportunity for future research.
Okay. Yeah, thank you.
We have another question from Travis. Travis, you want to ask your question verbally?
I can. So you mentioned the malting quality interactions that with MKK3 alleles. What have you found, and what are the implications for the different types of brewing?
Yeah, so I very briefly mentioned this, but our early malting quality mapping results in the S2-MET population suggest that the highly non-dormant N-star allele is associated with some grain modification traits like soluble over total protein, wort protein, and then free aminonitrogen. Those are really important traits, more so for industrial brewing. And by industrial brewing, I mean like Anheuser-Busch, Miller Coors, so Budweiser, Miller Lite, even things like Yingling. Whereas the craft malting profile is a little bit different. And we're thinking that pre-harvest sprouting resistance with the other two MKK3 alleles might not be compromising malting quality. And we also found malting quality QTL at non-SD2 loci, which is very promising for selecting for pre-harvest sprouting resistance and good malting.
Okay, well, I don't see any other questions in the chat box. I don't see anybody's hand up, so let's thank Dan for an outstanding exit seminar. And he'll be around for, I think, what, a couple more weeks, right, Dan?
Yep, definitely through May and then here and there in June. Okay.
Thanks very much, and thank you for attending Dan's seminar.
All right, thanks everybody.
Bye-bye.
This has been a production of Cornell University, on the web at cornell.edu.